So this is the starting point of our process. First thing is that we have a crushing machine here, and here we are going to put also your wastes there in Sweden, and we crush them here, and then we add water. And we try to get the dry matter content between 10 and 15%. That is good for the process, and that is also good for, or it goes through from valves and pumps, which we have here. So dry matter content about 15%. And then we pump the mass to the hydrolyzer, which is the place where we add enzymes. And then with enzymes, we suppose that the mass turns to the little bit better for bacteria. So sugars will be a little bit easier for them to use and so on. So we add enzymes and let it be there maybe overnight or something. We can adjust the pH also here and the temperature as we want. And when we have good amount of sugars there, then we move it to the reactor, which is the heart of our system. So that is a little bit forward, so maybe we go a little bit backwards to see that.
Yeah. It will be standing here for 24 hours in this container?
The time can be...
Depends on what it's...
Yes, it depends. For example, the stuff which we have used now, it's not very easy to get sugars out of it.
Okay.
But maybe in your case it might be easier, so it's maybe not so long time. But if we could say that during over the night, for example, would be the good.
Yeah.
And then from hydrolyzer we move it to the reactor, and here is the reactor, our bioreactor. And we can measure pH, we can measure oxygen levels, and here we can make the mass go round so that we can take samples from the circulating system. And we can, to the circulating system, we can also add some stuffs, for example base or acid, if we want to adjust pH. We can also make some other addings if we want by pumping stuff into this circulation. And when our masses are in, the pH is okay, we have sugars there and everything is okay, temperature is as we want, then we can add our bacteria, which are our workers, our real workers. And bacteria we grow here in these these glass fermenters. And to the fermenters we get the bacteria from the PMU, which is over there. So that is a system where we can aseptically grow those bacteria which we want in each case. And then to get enough amount of bacteria we have to use these, and then from these we pump our workers to the process.
And how do you grow the bacteria?
The system is same as in the PMU and as it is here. We can put there media for the suitable media for bacteria which we want to use, and then we lead gas, air if we want to grow aerobic bacteria, and nitrogen if we want to grow anaerobics. We can also make a mixture of gases. We have here a gas mixture and gas bottles usually in the behind of the system, and we can make the mixture we want. So for example, nitrogen, a small amount of carbon dioxide, and maybe a small amount of oxygen also if we need. So we can use for each of these places the gas mixture we want. For PMU, for these fermenters, and also in here in the process. And then when everything is there and bacteria has been added, the process will start.
How come it's a window here? What is the point?
We can see when we, for example, when we fill it, that where is the level, how high up it is, and it's also possible to see if bubbling is okay, and if it's foaming or not.
And also when, here you can see the level.
When the level is upper, then we can follow it from this window.
And what's the temperature in this one?
We can adjust the temperature. There's a water around this and we can have it not very high up, but for example between 20 or 60. The 40 degrees, 37 would be the best one quite often. So but the temperature we can adjust, and the pH we can adjust by adding base or adding acids to the pumping it to the circulation system. And then we take samples from those valves which are there. We can take samples, and we have there behind that door we have a gas chromatography, and we can analyze the ethanol, butanol, butanediol, for example, acetic acid. And then when we get the results, we can also drive this process. When we see what's coming out, we can change things when we know. It takes about 1.5 hours to get the results.
Okay.
And we also measure gases, the amount of the gases, the total amount of gas, and then methane, hydrogen, and hydrogen sulfur, and also oxygen. So there is a gas, there the gas analyzer. And then when we are ready, we can gather our stuff to the stabilizer, or if we don't want to take it there, we can also take it out from some valves. And after that, it's also possible to filtrate it, where we have a small filtrating unit there after all. So this is simply the system.
Very interesting.
And we are very eager.